Assalamualaikum and a very good day to Dr. Haryati. Today here we will be presenting our group presentation on the title of Structural Determination of Anti-Influenza Drugs in Combating Influenza Viral Infection. So before we proceed, here I introduce our team members, which is Lau Siu King, Nurina binti Arifin, Siti Nur Shikira binti Jamaluddin, and also me, Nur Ashikin binti Muhammad Rasul. So our topic focus more on influenza virus. So this is a structure of the influenza virus. So influenza a virus is the major cause of seasonal or pandemic flu worldwide. So the potential of pandemic influenza outbreak has attracted many scientists and researchers out there to find and create any possible anti-influenza drugs in preventing influenza viral infection. So currently we will discuss two types of protein inhibitor, which is neuraminidase inhibitor and fusion inhibitor to be specific cyclic P7 peptide as an anti-influenza drugs as strategy to overcome the viral infection.
Are representing about the structure determination of the neuraminidase and fusion inhibitor. So before I begin with this presentation, I would like to give a short summary on the process of the structure-based design. So this process are often required a multiple cycle before this optimized drug goes into the clinical trial. So the first cycle include with the cloning, purification, and also purification of our drug target. And we are going to do the structure determination of our drug target either by using X-ray crystallography, NMR, or homology modeling. And we can carry out a computer algorithm in order to position our compound into a selected region at the structure, okay, so that these compounds are scored and ranked based on their steric and electrostatic interaction with the target site, and the best compound will be tested with the biochemical assay. And in the second cycle, we are going to identify, okay, either our drug target and also our drug is a micromolar inhibitor in solution. If yes, then we are going to do some structure determination again, and then we are going to analyze, okay, our interaction, the interaction of our compound. So we are going to do some additional step, okay, in order to increase the potency by optimize our drug. And after the several cycle of optimization, we can see, okay, that there is an improvement in the binding and also the specificity of the target, okay. And next, I would like to discuss about the structure determination of our drug target, which is neuraminidase by using X-ray crystallography. So as we know, actually, this influenza neuraminidase has been recognized as a potential drug target since 1970 because it's highly conserved active, okay, which become an ideal for the target to be an target for the drug design, okay. So this NA is actually a tetramer, okay, for article polypeptide, and each polypeptide contain about 470 amino acid arranged in the 4 domain. So from this structure, okay, from this structure, we can identify that actually the positive charge consists of adenine 118, 292, 371 can actually interact with the carboxylic group of inhibitor, while the negative charge of these neuraminidase can interact with the guanidine or amino group of the inhibitor. And we also identify that the presence of a 150 loop in the residue 147 until 152 actually adjacent to the active site, okay, which help for the binding of the inhibitor. So next, okay, I would like to discuss about the NMR analysis, okay, for our drug, which is oseltamivir with the neuraminidase. So we are going to use some a model membrane protein. Model membrane model membrane, which will be prepared by using the MPC. So this NMR experiment will be recorded on the Bruken Advance 500 megahertz spectrometer, and we also going to do 2D noisy experiment in order to identify the interaction of this drug and our target drug. So we are going to do the NMR experiment in the spectra by using carbon 30. This is the NMR analysis. So we can see, okay, there are about 3 spectra here, okay, and we use the carbon 30 NMR. Okay, so here the first one is actually the DNPC, okay, which means that only the model membrane, okay, and the second one is only our inhibitor, which is the oseltamivir, or we call here as a GS4104, and the third one will be our combination of the oseltamivir and also the model membrane, which is the MPC unilamellar vesicle. So based on this result, okay, we can actually put into a 3-category signal, okay, which are the one that remain sharp but are shifted. Then we also have the broad and shifted, and finally those are broad beyond detection. So this differential broadening of the GS 4104 resonance arise because of the different part of the drug bind to the lipid bilayer with a different affinity. So next, we have here the 2D noisy spectrum of the GS4104 incorporate with the DMPC unimelarosica. So such spectrum can provide us information of the intermolecular interaction from the intermolecular noise between the two molecules. Okay, and this noise actually can arise when the two atoms either within the same molecule or a different molecule are at distance less than 5 Armstrong. Okay, so based on this result, okay, we can observe, okay, actually that our GS4104 or oseltamivir actually use it hydrophobic chain and also the cyclohexane group to bind via the hydrophobic interaction with the lipid spanning both of the head group as well as the interior of the lipid bilayer. So we can conclude that the oseltamivir actually can interact with the membrane fusion to inhibit the NA activity. And the membrane fusion is actually the process in the biological system and involve the union of the two opposite bilayer in order to complete a process such as exocytosis or viral infection. And these oseltamivir have interfered the cell-cell fusion, thus interfere with this virus entry step. Another drug that we can use actually to be as an anti-influenza drug, okay, we have also the Abidol, okay, the Abidol, which is the fusion inhibitor. So basically, based on this result, okay, we can see that we have two spectra. The first one would be the spectra for the Abidol in the hard water, and another one would be the Abidol in the with our modern membration, the MPC also. So based on this result, we can see that, okay, the Abidol interaction with the lipid induce chemical shift changes in the Abidol resonance when we compare to the observed in the water, which means there is some interaction. So this albedo can be an efficient inhibitor.
Next, I will further elaborate on the structure of the two types of protein that are used in existing on design of drugs for treatment of disease. So first protein is neuraminidase. So neuraminidase is a type of protein, or also called as Na, that is a tetramer of 4 identical polypeptides that exist on variant surface. And the 4 monomers fall into 4 distinct structural domains that consist of head, stalk, transmembrane, and also cytoplasmic tail. So this is the structure of the neuraminidase. So we go through first to the structure of the Na itself. So the catalytic head of NAs consists of a box-shaped structure with 4 monomers, and each monomer is in the form of a 6-bladed propeller structure, with each blade having 4 antiparallel beta sheets that are stabilized by disulfide bond and connected by loops. So a functional catalytic site is present on the surface of each monomer, and it arranged sideward rather than upward. This is to promote the ability to cleave the sialic acids from nearby membrane glycoprotein to prevent the virus trapping. So the active site is located in a deep central cavity in each of the 4 monomers that consists the active form of the neuraminidase. The active site is so important for virus spreading and is highly conserved, making it desirable target for neuraminidase inhibitors that can be used in antiviral therapies. So next is the transmembrane domain, which the N-terminal hydrophobic transmembrane domain attaches the Na to the viral envelope. So this attachment provides the signal for translocation from the endoplasmic reticulum to the apical surface, as well as the association with the lipid raft. So actually, both the cytoplasmic tail domain and the transmembrane domain contribute to the transport of the glycoprotein to the apical plasma membrane. So further with the stalk domain, so the cysteine residues at the neuraminidase stalk may assist with the tetramer stabilization by enabling disulfide bond to form between each monomeric Na unit. And the presence of the carbohydrate side chain within the stalk is thought to contribute further to the stability of the tetramer. So basically, the length of the stalk region can have a significant impact on particular virus characteristic. So previous study has proved that reduced stalk length will impact the Na activity of the variants because the reduced height may hinder access to cellular sialic acid expressing receptors, and that the towering HA blocks the shorter Na catalytic domain from gaining access to the sialic acid. So the lack of the Na function would trap the viral particle and prevent its attachment to the cell surface receptor for viral entry. So reduced stock size may also alter the geometry and dynamics of the enzymatic pocket itself, modifying its affinity for salicylic acid as shown in this figure on the next slide. This figure shows the part A, the optimal NA stock length enable the NA catalytic head access to the salicylic acid expressing receptor, while for the figure in B shows that the reduced stock length may impact the ability of the NA to contact salicylic acid on the cellular receptor, as the neighboring HA may sterically hinder the shorter neuraminidase catalytic domain from gaining access to the salicylic acid. So done with the neuraminidase protein. So I continue with the second type of protein, which is cyclic peptides, or to be specific, we have discussed on cyclic P7 peptide in the next few slides. So this is the figure that shows the structure of the cyclic P7 peptide. So generally, the cyclic peptides are usually composed of 5 to 14 amino acids and have a molecular weight of about 500 to 2000 Dalton. So the moderate size and diverse functional groups of peptides ensure that the contact area is large enough to provide high selectivity and their potential to form multiple hydrogen bonds that can lead to strong binding affinity. So in addition, the cyclization of the peptides also facilitate the passage through the cell membrane, thus broadening the potential use of cyclic peptides in generating the structure and also the function features that can be used as the pharmaceutical agent. So because of that favorable characteristic features of that cyclic peptide, various cyclic peptides have been exploited for biomedical and other purposes that we'll be discussing more in the next few slides.
I will proceed the presentation with the importance of the reported structure in the design of drugs for treatment of influenza A. This figure shows influenza A virus. And my part of presentation will be focusing on neuraminidase. For your information, neuraminidase is one of the two major surface glycoproteins found on the influenza virus particles. Its function is to cleave off the terminal sialic acid from the cell surface glycoconjugates, which is the receptor for the other surface protein, hemagglutinin. It is known that inhibition of neuraminidase can effectively delay the release of progeny virions from the surface of infected cells. Therefore, there is a drug development targeting the neuraminidase protein, as it is proven to be a useful target for antiviral therapy because its active site is highly conserved in both amino acid sequence and spatial orientation, as well as it is prominently located on the surface of virion, so it will be easier to be targeted by the neuraminidase inhibitors. So this figure shows the interaction of the neuraminidase active site with sialic acid in the absence of neuraminidase inhibitors. Active site can be divided into 5 subsites, namely from S1 to S5. So S1, which is made up of arginine 118292371, will be able to interact with the carboxylate side chain of sialic acid. O4-hydroxyl can form interaction with residues glutamic acid 119, aspartic acid 151, and glutamic acid 227. Oxygen and nitrogen atoms of the acetamidol form hydrogen bonds with arginine 152, whereas the methyl group lies in a hydrophobic pocket near isoleucine 222 and tryptophan 178. O8 and O9 hydroxyl groups of the glycerol side chain are hydrogen bonded to the residue glutamic acid 276 and 277 to make an important contribution to the overall binding. So by studying well the interaction of the neuraminidase active site to the sialic acid, so there is a development of the neuraminidase inhibitors to act as a competitive inhibitor to stop the neuraminidase activity from interacting with the sialic acid. So there are 4 main examples of neuraminidase inhibitors lead to the drug development. So namely zanamivir, oseltamivir, peramivir, and laninamivir. So for zanamivir, oseltamivir, and peramivir, they are the derivative of the earliest transition state analog of the neuraminidase substrate 2,3-hydro-2-deoxy-N-acetyl-neuraminic acid DANA, whereas laninamivir was produced by a modification of zanamivir. All of them mimic the substrate of neuraminidase, which is the sialic acid, and they can bind to the neuraminidase with higher affinity as compared to the sialic acid. So as we can see from this figure, so the development of these 4 types of inhibitors are actually based on the sialic acid because they have the basic structure similar to the sialic acid. And then those that are highlighted in red are the side chains or functional groups, which is different from the sialic acid. And those side chains which makes the inhibitors to be able to have more interaction and higher binding affinity to the neuraminidase. So for zenamivir, it has a positively charged guanidino group replacing the hydroxyl group that is linked to C4. So it increased the binding to the neuraminidase approximately 10,000-fold as compared to sialic acid. For oseltamivir, C4OH and glycerol side chain of DANA are replaced by amino group and pentyl ethyl ester group respectively. For peramivir, it is a cyclopentane derivative with a guanidino group and pentyl ethyl ester group. For your information, cyclopentane is known to exhibit potent and selective inhibitory effects, especially on influenza virus neuraminidase. And for laninamivir, it is actually the modification of zenamivir where methylation occurs on the C7 hydroxyl group of zenamivir. So neuraminidase inhibitor structures are developed based on DANA or designed to mimic its binding to the neuraminidase, but differing structures results in interactions with different amino acid. So all of these inhibitors with the negatively charged carboxylate group are able to form strong hydrogen bonds interaction with the subsite 1 that consists of arginine 118-292-371. Guanidino group, which is only present in the neuraminidase inhibitors like zenamivir, peramivir, and laninamivir, they can form stable hydrogen bonds and strong electrostatic interactions with the acidic groups of glutamic acid 119, aspartic acid 151, and glutamic acid 227. Glycerol side chain are hydrogen bonded to the residue, glutamic acid 276 and 277, to make an important contribution to the overall binding of neuraminidase to the inhibitors. Oxygen and nitrogen atoms of the acetamidor can form hydrogen bonds with arginine 152, whereas the methyl groups lies in a hydrophobic pocket near isoleucine 222 and tryptophan 178. So as shown in this figure, we can see that neuraminidase inhibitors have more interactions and higher binding affinity to the neuraminidase as compared to the sialic acid. Therefore, the presence of neuraminidase inhibitors will act as competitive inhibitors to the neuraminidase. Neuraminidase inhibitors, which mimic sialic acid structure in its transition state during Na-catalyzed hydrolysis, represent a class of effective anti-influenza drugs. So as I just explained, they are zenamivir, oseltamivir, peramivir, and laninamivir. So for zenamivir, it is mainly delivered through inhalation and was licensed worldwide since 1999. For oseltamivir, it is mainly administered through oral and was licensed worldwide since 1999. For peramivir, it is an intravenous formulation that is developed only for compassionate use and was licensed in few countries like Japan, South Korea, China, and USA since 2014. For laninamivir, which is the modification of zenamivir, was only licensed in Japan since 2010 and is currently in phase 3 clinical trials in USA. As a summary, after budding, virions need to detach from the host cell by viral neuraminidase, which possess the sialidase activity, to cleave off the sialic acid, thus releasing the virion's progenies. So this step is the target of the anti-influenza neuraminidase inhibitors that are used in clinical practice. So in the presence of neuraminidase inhibitors, it can stop the release of viruses from the infected host cells and prevents the new host cells from being infected. Thus, it can suppress the viral population and allow the time for the host immune system to eliminate the virus and eventually control the infection.
Good day to Dr. Haryati. So today I will be presenting on the importance of fusion inhibitors and the ways they could be incorporated when designing drugs which can actually combat the influenza A virus, which has been a very upcoming conundrum across the globe, which really requires a very insistent and eminent sort of treatment to this, which the research community is currently working on. So before I present, I would like to give some key points for whatever that I'm going to present today, a light brief highlights. So first of all, we will look into the viral mechanism, the particular machinery, which actually how it really propagates throughout the cell and how it replicates and so on, and which machinery has been targeted by the scientist community on the whole by the fusion, by using infusion inhibitors, and how do they incorporate them into drugs, which sometimes have the potential to become successful drugs, and also the drugs which have been successfully established amidst the market. So first and foremost, as I said, the viral hemagglutinin machinery as an antiviral target. So this particular machinery has been targeted as an antiviral mechanism by the research community themselves to provide medicines, provide drugs to combat the influenza A virus on the whole. So let me brief. First of all, these viral hemagglutinin proteins, these particular proteins, they tend to have this particular domain that bind to the glycans that are found in the cell surface of the sialic acid, which literally happens over here. So when they bind, what happens is internalization of the virions via endocytosis will lead to the decrease of the pH, which means the entire environment tends to be acidic amidst the endosome. So this is the turning point, or this can be marked as the climax, which actually leads to the subsequent effect or the tailing effect, the repercussions, which is caused by the virions, which actually tends to lead to the final replication, because this particular acidic environment could lead to the irreversible conformational change of the HA protein. So when there is a conformational change, the fusion protein, the fusion peptide of this particular HA protein is exposed. When it is exposed, this triggers the fusion with the endosomal membrane of host cell, and thus the viral ribonucleoproteins are released into the cytosol, they migrate to the nucleus for subsequent replication, and the entire cell is damaged, done by the virus itself. So scientists have targeted two important factors here. One is blocking of the HA binding to the sialic acid residue, which is the beginning or the initiation of the entire process itself. Or another target could be the blocking of the HA conformation change, which roots out from the acidic environment of the endosome itself. Hence, this is particularly the rough idea of what really happens due to the HA mechanism of the virus itself. So the internalization of virion could increase the acidity in the endosome, which will lead to the conformational change, and thus the repercussions would be the membrane fusion followed by replications. So what do scientists do literally, and what do they have discovered is that this particular fusion inhibitors, how do they function? Now they function by preventing this acid-triggered conformational change, which then enables them to incorporate these fusion inhibitors when designing drugs as the antiviral drugs to combat influenza A virus. So the first fusion inhibitor, which I'd like to talk about, is P7 cyclic peptide, which is currently on the stage or the status of being a potential fusion inhibitor to be incorporated into drugs to combat influenza A virus. So from a very pivotal anecdote, P7 cyclic peptide is well prominent, well eminent for its specificity. Why? Because the structure of this peptide is solely conjectured on the note of the heavy chain complementarity determining region loop of BNEB F6V3 and framework region 3 of CR9114. So these components in this particular cyclic peptide makes it more specific, which makes it to be highly eminent and a highly potential candidate to be incorporated for drugs that are actually against influenza A virus. So looking in depth, note on a very in-depth sort of deliberation, P7 literally binds to the HA stem epitope. So this is where it hits the key point, the climax, where it blocks the low pH, the acidic pH, which is actually the prime factor which causes the conformational arrangements, followed by the membrane fusion, followed by replications. So this is particularly the first step in which the P7 cyclic peptide attacks or combats. It combats the low pH induced conformational rearrangements first. It blocks the entire process by its specificity, by binding to the highly conserved HA stem epitope. So what makes it P7? What makes P7 super specific for group 1 HAs? It's because of the glycosylation of ASN38 in group 2 HAs and substitution of tyrosine 49 group 1 for a larger ASN group 2 that improves binding. These groups are literally which belongs to the HA itself, two different types of HAs, okay? So this particular figure here, it shows the structural elucidation. of the group 1 HA specificity of the peptides. Moving forward, Arbidol. Now, Arbidol is one of the most successful fusion inhibitor, which has been incorporated into drugs successfully amidst the market to prevent influenza virus, to combat influenza virus, and it's one of the most successful feats by the scientist community themselves. So applauses to them. Well, when we come to Arbidol, this particular topic, literally we will be focusing more on the hydrophobic cavity found in the HA trimer. This hydrophobic cavity in the HA trimer would be literally targeted by this particular Arbidol. So Arbidol works by actually targeting the hydrophobic cavity in the HA trimer, which is the particular place where it binds. So this particular binding literally increases its specificity on greater levels, on greater heights. So where does it bind? Let's look into a closer note. Arbidol consists of peptides that function as fusion inhibitors such as F1, F5, F7, and F8. So all these components all these peptides, they tend to bind in the hydrophobic cavity in the HA trimer stem at the interface between two protomers. And binding of this Arbidol to the cavity, hydrophobic cavity in the HA trimer will literally, it will make some hydrophobic interactions for sure, and it will induce some conformational rearrangements to form a network of inter and intraprotomal stall bridges, which will then subsequently or concurrently, it will literally block the pH-induced conformational rearrangements. So it will block the entire network by causing a different sort of conformational rearrangement, which is the formation of this particular intraprotomal stall bridges. So by that, it stabilizes the perfusion conformation of HA that inhibits the large conformational rearrangements, which will lead to the membrane fusion. That's the functions of, those are the functions of Arbidol. So Arbidol has a very high shape complementarity with the binding sites in these two groups of HAs, group 1 and group 2. It has a very high complementarity due to the presence of F7 and F8 that work as a minor fusion inhibitors. So all these structural informations, all these opulent informations on Arbidol has really enhanced or enabled the scientist community to come up with superfluous drugs that can combat influenza A virus on a very high note. So incorporation of these sort of fusion inhibitors are really imminent and really significant, and the scientist community is still conforming with its efforts going forward and progressing with its efforts by literally researching more in depth on the structures of Arbidol, P7 cyclic peptide, and many sorts of other fusion inhibitors to be incorporated into the drugs and to come out with really efficacious drugs to put an entire full stop to the influenza A virus, though it might sound a little, well, perhaps not necessarily impossible, but let's just hope for the best and let's save our globe from this particular influenza A virus. Thank you.